This is a video showing some animal activity observed by researchers conducting a preclinical animal test using Lucid-MS with some data points. Quantum BioPharma is trying to illustrate some of the potential of Lucid-MS for the potential treatment of multiple sclerosis. Scientists conducted extensive research over the past 11 years, evaluating several compounds in the test tube and in animal models. In this test, Lucid-MS exhibited positive activities in an animal used to test activities against MS like disease symptoms. Here we present one of the examples from many such studies conducted by the same researchers. And part of the reason we believe our drug candidate, Lucid-MS, has the potential to affect functional recovery in multiple sclerosis. Our researchers used multiple animal models in their research. Here we show one of the animal models called the EAE model that shows inflammatory symptoms as well as loss of myelin in the brain, leading to loss of function. This shows the graph and video of a deceased mouse that has not been treated with Lucid-MS. You can see that the hind legs and tail of the mouse are still paralyzed after the experiment is completed. In autoimmune and neurodegenerative disorders like MS, the myelin sheath around the nerves gets damaged. Plaques are seen in various regions of brain and spinal cord, and simply put, there is a loss of signal between neurons. This next video was recorded in the early days of treatment with Lucid-MS on the third day of treatment. The clinical score for the mouse was 3, and characterized by flaccid tail and completely paralyzed hind limbs. This is a high score, and a lower score indicates a healthier condition. Treatment was continued, and health was monitored for days and weeks to come, as seen in this graph. The symptoms start to recede. Intermittent ups and downs are noted between scores 1 and 2, but a gradual reduction in symptoms and improvement in functional recovery was observed on day 42 of the treatment on the same mouse with Lucid-MS, which is 57 days since the beginning of the disease. The mouse mostly recovered with minor symptoms, such as a limp tail at the tip. The clinical score at this time was about 0.5. Characterized by mild signs of the disease. After a couple more days, the mouse appears to recover completely and functional recovery was observed. These two videos of the same mouse or two illustrate the effect of the drug in one preclinical animal model used for the evaluation of efficacy when developing drugs for multiple sclerosis. The above mouse was part of a larger cohort study. Our researchers thoroughly investigated Lucid-MS with peers to try to confirm the potential of these compounds and whether to enter further advanced studies in clinical trials. Lucid-MS prevented the loss of myelin and promotes remyelination. As a result, we believe the disease is prevented and we see functional recovery in these animals.